All right, grade 12s, we are ready to wrap up chapter 18. That's the good news. The bad news, of course, is there's a test coming up on it. What we need to do is talk about some other non-Mendelian situations when it comes to our genes. So let's start off by talking about selective breeding and gene interaction. So for years, humans have been trying to get the most desirable traits out of animals and plants. And of course, that's a form of artificial selection, otherwise known as selective breeding. And essentially, humans just take the most desirable trait from the offspring that they see and then just keep breeding when they see the desired trait that they want. So when we want to keep similar phenotypes, that's exactly what we do. We select related organisms at times, right? So sometimes it's parents and offspring, or offspring and offspring of the same breeds or families. Of course, that leads to inbreeding, which can work okay with plants and animals, but nothing that we would really want to do with humans, because what ends up happening is if there are defective mutated genes, then you will simply increase their frequency in that population, in the gene pool, and then you're more likely to see irregularities or issues or problems in that case. Now let's talk about something called a polygenic trait. So this is when we have one trait, but it's going to be controlled by several pairs of independent genes. So many genes, polygenic, controlling one trait. Sometimes it's additive or sometimes it's subtractive. So let's look at skin color for an example of a trait that's affected by at least two genes. And we're going to look at a fairly simplified ratio. Of course, we wouldn't really expect that 9:3:3:1 ratio like we would see with two genes, because there's only one trait being expressed. So if we looked, and this is very simplified, skin color is much more complicated than we're going to look at here. So let's just say there were two genes, an A and a B gene, and if you have the more dominant alleles that you have, the darker your skin is going to be. So we can go from as fair as quite white skin, having the homozygous recessive for both genes or both alleles, and then you can see that as we add more dominant alleles, the darker the skin color is. So there you go. We've got more than one gene affecting one trait, polygenic. Now this does lead to something we call epistatic genetics. This is when one gene can affect the expression of another gene. So we're going to take our common mouse example, and let's look at the coat color for mice. So it involves two genes. The first gene leads to the expression of a black or brown pigment, which we're going to use B, black being dominant, so capital B for black, little b for brown. But then there is a second gene which is epistatic. In other words, it can control the expression of the B allele. So we can call it C for color. And if you have the dominant capital C, then the color of the coat will be expressed. So we will see that black if there's a big B allele, or we'll see the brown if there's the little b in the homozygous recessive condition. However, if you have the recessive C allele, the little c allele, homozygous of course, because it's recessive, then there's no expression of pigment. You wouldn't see any color at all. So we have some examples of genotypes at the bottom of the screen here. So if you have both dominant alleles, so a capital B and a capital C, it doesn't matter what the other alleles are, then we're going to see a black color. Big C means we're going to see color, and the big B means a black color. The second scenario is we're homozygous recessive for the little b, which is the brown allele, and we still see a big C. So yes, we're going to see color, and in this case, it's going to be brown. Or it doesn't matter what B alleles we have. If we see two little c's, we don't have the dominant color allele, so we don't see the color expressed. And in this case, we would have white or albino mice. Now, the next slide is going to allow us to see this done on a Punnett square. So let's say we had two black mice that are heterozygous for both alleles. So you know this is going to give us a 4x4 Punnett square with all those different possibilities of alleles in the gametes. And when we complete the Punnett square, we can see that any mouse that has a big B and a big C is black. Mice that have the little b's, two little b's, but they have at least one big C are brown mice. But here we don't have that dominant capital C that allows pigment to be expressed. So we have no color in these mice. So you see we don't have the 9:3:3:1 ratio we would normally expect, right? We have a 9. Do we have 9? 1, 2, 3, 4, 5, 7. Yes. 9 black mice, 3 brown mice, 4 white mice. We don't even have a 4th phenotype to worry about. There's only 3 phenotypes in this situation. So another example would be in dogs. Let's say there's a gene B, again, that produces a black color, and a little b that produces brown, just like the mice. And we have a gene W, but this time the capital W prevents expression of any pigment, thus producing white coats. The little w allows for expression of pigment. So what will be the offspring if a white dog, so remember that capital W causes no pigment, so a little different than the mouse example, is crossed with a black dog. See, so again, we're going to see pigment expressed. There's our black allele. All right, so we would set up our Punnett square. Now, folks, we only have to do a 4x2 Punnett square because you understand that being completely heterozygous here, this dog provides 4 different possibilities for alleles in the gametes, whereas this dog, there's only 2 possibilities. So when we fill in our Punnett square, we get our results. So we would see that this would be a white dog because remember that a capital W means a white dog. Doesn't matter what the B alleles are doing. So white dog, black dog, black dog, white dog, black dog, and 1 brown. All right, so we see 4 white dogs, 3 black, 1 brown would be the phenotypic ratio for that cross. So that's an example of what we call epistasis. Think of it as sort of an interference. That's how I remember it. Stasis, or it sounds like static, epistatic. Static is interference, like if you were searching for a radio station. In the old days, when we actually had to turn a dial to get to a radio station instead of just pressing a button or just going to the next available signal, we would have static because there would be interference going on so that you couldn't get your radio stations. It's kind of, this is a form of interference of one gene on another gene. Okay, let's talk about another situation called the Pleiotropic gene. This is kind of the flip of polygenic. So in Mendelian genetics, remember that there's one gene with 2 possible alleles to govern that one trait. In the case of a pleiotropic gene, there is still just one gene, but it actually affects many different traits. So it's non-Mendelian. Let's look at an example, Marfan syndrome. So Marfan syndrome is caused by one defective gene that codes for connective tissue in the body. It's expected that Abraham Lincoln had Marfan's syndrome. Somebody who have Marfan's is often very tall. You have an enlarged heart, so you have a propensity for potential heart problems like an aneurysm when you get older. But because connective tissue is so pervasive through many, many systems of the body, that one defective gene can affect so many different things going on in your body. And there's a great long list there, and you can look at that for yourself later. Sickle cell anemia is another very common example. You see this on lots of test questions. it is also an example of a pleiotropic gene. So there is one gene that codes for your hemoglobin. So remember, hemoglobin lives in your red blood cells, and its job is to carry oxygen. And so there are two alleles, one that creates normal hemoglobin, but then a mutated defective allele that creates abnormal hemoglobin, which actually leads to a sickle-shaped red blood cell, which you'll see a picture of that in a second if you're not exactly sure what a sickle is. All right, it's a field farming instrument. Now, if you're heterozygous, so if you had one of each of these, then you would be called a carrier because you're carrying the defective gene. In this case, as I go to the next slide, so let's continue. So again, you're being reminded that the red blood cell shape, so here's the sickle cells right here. Okay, so that's what a sickle looks like. These are our nice normal red blood cells. Well, they can't carry oxygen very effectively. Plus, they have issues with the length of their life, as well as they could actually, because they're more jagged instead of smooth, they can actually make tears in your blood vessels. And so this one effect causes all sorts of issues in the body. Now, if we looked at a cross here between two parents that carried that mutated sickle cell gene, they would create this result in their offspring. We would have one normal child, so not affected by sickle cell in any way. Then we would have two carrier children, like the genotypes of their parents. And then we would have one child with full-blown sickle cell anemia, where essentially every red blood cell has a sickle shape. Now, this will come up on the slide later. If you are a carrier, about 65% of your red blood cells are normal, and 35% of your red blood cells are sickle-shaped. So you've got enough good red blood cells to do the job. And again, now another trait that's affected by this one gene is that you are protected against malaria. So malaria is a blood infection, and it just so happens that you would be protected against malaria. So that's kind of an advantageous benefit of that mutation if you happen to carry the sickle cell allele. Again, this is another slide essentially showing the same thing, and here we have the stats on having the sickle cells and how many sickle cells you have. Now, just as a point of interest, I have a little list here about, well, how many of your traits are affected by this one gene. So if you had sickle cell anemia, you would have fatigue, because of course you're not carrying enough oxygen on a regular basis, so you're tired a lot of the time. You end up with pain, you end up with swelling of your hands and your feet, you're subject to many more bacterial infections, you can have pooling of blood in your spleen, the liver congestion, lung and heart injuries, leg ulcers, eye damage. So you can see that one gene can affect many traits or things that are going on in your body. So let's recap a lot of what we've talked about in chapter 18 here. So typical Mendelian genetics on the far left here, one gene with two alleles, so two forms of that gene. Remember, the gene is just the segment of DNA on your chromosomes that it's coding for a particular trait. And so only one trait, one gene, so that's very Mendelian. There's two different phenotypes that could be expressed because you've got that principle of dominance where you have the one dominant gene, so you have the dominant trait, or you have the two recessive alleles, and you will show the recessive trait. So let's compare that to multiple allele situation. So we're still only talking about one gene, but now there's more than two forms that gene can take, more flavors than just two. We're still only going to govern one trait, but now we have the potential of three or more phenotypes. So remember, with blood types, which is a multiple allele situation, you can be blood type A, blood type B, blood type O, AB, all right? And then it's a case dependence on whether there's any dominance or not. Sometimes there is, and sometimes there isn't. Okay, incomplete dominance. So remember, with incomplete dominance, we're still looking at one gene, we only have two alleles, we're still only talking about one trait, But this time, because there's no complete dominance, remember we can get that sort of blending situation like with the snapdragons. We can get that pink showing up from white and red, so that's 3 phenotypes. Or we can actually get codominance, where both forms of the gene or the alleles are expressed, like with the roan cattle, like the black, or blood types A and B. That's an example of codominance. So we have at least three phenotypes. There's no true dominance. So today we talked about polygenic traits. So this time there's polygenic, many genes controlling one trait. So there's still 2 alleles. We're only dealing with 1 trait. There's less than 2 phenotypes per gene. Remember how that's how come we won't have the 9-3-3-1 scenario. All right. And then again, it's case dependent about whether there's true dominance or not. And then lastly, it's the pleiotropic gene, where again, it's 1 gene, there's only 2 alleles, but 2 or more traits are affected by that 1 gene. Okay, and again, case dependence on the dominance. So that, ladies and gentlemen, is the end of chapter 18. Happy studying. I hope you do very, very well on the test coming up a little later.